everyone, I am Dr. Sayani, Assistant Professor of Biology. Today we will discuss about the nucleic acids, the DNA and RNA, which is deoxyribonucleic acids and ribonucleic acids, which are the essential genetic materials for all living organisms. What are the nucleic acids? So nucleic acids is a biopolymer which is made with small nucleotides, which is crucial for the life processes. They store all the genetic informations. DNA and RNA are the two types of nucleic acids. Yeah? DNA and this is the RNA. This is a double helix structure and DNA has double helix and RNA is a single stranded. Structure of DNA. DNA consists mainly it's a double helix forms. It has 4 different nucleotides: adenine, guanine, thymine and cytosines. It has a sugar phosphate backbone. So this is the structure. This is the phosphate groups. This is the sugar deoxyribose. It is a pentose sugar. You can see there is 5 carbon. 1, 2, 3, 4, 5. And this is the nitrogen groups. It depends on A, T, G, C. It varies. The nitrogen bases. There is adenine, which is called as the purine, and guanine, which is also a purine. Cytosine and thymine are the pyrimidine. So this is the structure of adenine, this is the structure of guanine, structure of cytosine and structure of thymine. DNA double helix. So DNA double helix occur due to the base pairing. So for the base pairing, the 2 nucleotides join together with hydrogen bonds. You can see in between G and C there are 3 hydrogen bonds. So and in between T and A there are 2 hydrogen bonds. So in between G and C or C and G there are 3 hydrogen bonds. In between A and T or T and A there are 2 hydrogen bonds. Replication of DNA. In our cell, DNA replicated means doubled from 2 to 4, 4 to 8, because for cell growth and development replication is required, means multiplication of DNA is required. So the conversion of 1 DNA to 2 DNA, 2 DNA to 4 DNA is called as DNA replication. Then transcription. In central dogma of life, DNA is transcribed into RNA. So then RNA will be converted into protein in translation process, and this totally occurred for passing the genetic informations. Here this is the DNA, and from DNA RNA will be produced. And in RNA there is one change. The nucleotides are adenine, uracil, cytosine, guanine. Means thymine is not present. So T will be replaced by U. Types of RNA. There are mainly 3 types of RNA. Though some other non-coding RNA are present, but main 3 types of RNA. One is mRNA, which carries the genetic code from DNA to ribosome. So this mRNA actually codes for the protein. Another one is the tRNA, which is called as transfer RNA. mRNA is a messenger RNA, tRNA is a transfer RNA, and rRNA is a ribosomal RNA. So tRNA transports the amino acids during the process of translation to the ribosome for protein assembly, and rRNA forms a part of the ribosome structure, catalyzes protein and catalyzes the protein synthesis. So the role of RNA at mRNA, which actually provides a blueprint of protein. I already told that the mRNA will be transcribed into protein, translated into protein, and the tRNA which brings the amino acids, and rRNA forms the peptide bonds. So tRNA brings the amino acids and the amino acids join together to make a polypeptide or protein. So the differences between DNA and RNA. DNA is double helix, or it has the double stranded. RNA is single stranded. It has in DNA there the nitrogen bases are adenine, guanine, thymine and cytosine. In RNA there is adenine, uracil, cytosine and guanine. DNA is more stable because it is double stranded. RNA is less stable because it is single stranded. The sugar in DNA is known as deoxyribose and the sugar in RNA is known as ribose. So DNA is required for the genetic storage and RNA is mainly required for the protein synthesis. Thank you.